Apologies for the layoff, lads. I was fighting the Avengers. So, let's do some biochemical tests. Now, the thing with this topic is very, I say easy, but there's a bit to remember. So, don't be a pussy. Let's get this one done. So, what am I yimming and yamming about when I say biochemical tests? Well, AQA, the big villain, they'll give you a sample, okay? And they're like, yo, bro, what's inside this sample? And you need to be able to use your knowledge of biochemical tests to figure out what's inside the sample. And we'll go through all of that in this video. So subscribe the video, like, all that bollocks. Let's get into it. So here we've got 3 different carbohydrates of deviating lengths of different sizes, okay? You've got the midget here, you've got the small, medium one here, and you've got the big bro. So in this video, we're going to be mainly covering how to test for the monosaccharide and the disaccharide. So the monosaccharide being the midget, and the disaccharide being, still a midget, but, he's got a friend, right? So here we've got a monosaccharide. And we call these bad boys a reducing sugar. Now, what am I yapping about when I say a reducing sugar? Well, when I say a reducing sugar, and this means that it has the ability to give something an electron, okay? That's what reduce something means. Let's say if I was to reduce you, hey bro, I want to reduce you, okay? That means I want to give you an electron, right? I'm so nice. And that's what these sugars can do, these monosaccharides do. Most of these guys, almost all of them, they have the ability to reduce things. They have the ability to give other people electrons. And because they're so generous, we can test for that, right? We can take a solution, throw it at these monosaccharides, and this monosaccharide will reduce that solution that we've given it. And some solutions actually show this. Let's say a solution has been given an electron. Sometimes if you pick the right solution, it will change color. And that's what we can do here. We take something called Benedict solution. And that's the wrong Benedict. That's Benedict Cumberbatch. I mean the Benedict solution. This thing right here. This thing will change color when you give it an electron, okay? It's kind of like, I don't know, he's no, that's weird. It's like he's so happy that you've given him an electron that he changes skin. No, that's still weird. He changes color when you give him an electron, okay? So that's what you should understand about Benedict solution. When you give it an electron, it changes color. And that's the thing that's how we test for these monosaccharides. When you put Benedict solution, you combine it with Benedict solution, and you always have to heat it up. And when there is a presence of monosaccharides, it will reduce the Benedict solution, causing it to change color. And there's a spectrum, okay? Depending on what kind of color it changes to will tell you how concentrated, how much monosaccharides there is. So here I'm showing you this spectrum here, and it goes from lowest concentration, or nothing, to the highest concentration. So blue means nothing, there's none. Green means, there's a little bit of monosaccharides in there. Orange means there's quite a lot, right? And here, brick red means there's so much, okay? And so just download this thing into your brain, understand it, learn it, memorize it. Pretty easy. AQA does ask you this in the exam. They're like, yeah, we've got a solution, it changed this color. What does this mean? And I'll show you a problem here, okay? This is a problem that, some random guy just did. Okay, I don't know him. And it has a bunch of colors. So why don't you try and tell me how many monosaccharides are in each of this solution? Try and tell me. So here we've got red, and that means there's quite a lot of monosaccharides. Here, blue, that means there's nothing, okay? Orange, that means there's quite a lot in here, okay? Blue again means nothing. And red here, it means there's quite a lot. And blue usually means that it hasn't really changed color, okay? If it hasn't changed color, that means there's no monosaccharides in it. And that's what Benedict solution is. It's blue. That's what Benedict solution looks like when it hasn't really been tampered with. Naturally, it just looks blue. And so when it stays blue, that means nothing's really changed. But when it changes to red or whatever, that means it's been reduced. So pretty easy. Now, some of you nerds might have noticed a problem with this. And there is a problem. The problem with this whole thing is that it is semi-quantitative, meaning just because it changes color doesn't mean it tells me the number, the exact value of how many monosaccharides there are in the solution. So just because it turns, I don't know, it turns green, right, which means there's some monosaccharides in it, it doesn't tell me the exact proportions, the exact value of how many monosaccharides there are. And in order to combat this issue, in order to fix this, we use a colorimeter. Now, what is this? Well, a colorimeter is basically this little machine, and it shines light through the solution, and depending on how much light went through, it will tell you how concentrated the solution is, and it will give you a value, okay? Now, you don't need to know too much about colorimetry just yet, okay? Don't worry about that. But just understand that if the exam asks you, like, how you can quantify glucose, how you can, I guess, know exactly how much glucose there is in a particular solution, you can say colorimetry, or you can also say a scale. You simply just take the solution, you filter out the precipitate, you weigh it, and boom, that's it. So you already know how to test for the monosaccharides, right? All you do is you put our boy Benedict in it, and it'll tell you the color change, and that's pretty much it. But how do we test for the other midgets, okay? The slightly bigger midgets, right? The disaccharides. How do we do that? Now, the thing is, a disaccharide is a non-reducing sugar, meaning he's not a very nice person. He's not a very nice individual, so he doesn't exactly donate electrons to other people. So how the hell do we do this? So because he won't give the Benedict solution any electrons, if I was to put this in Benedict's solution, it wouldn't change color because it hasn't been given any electrons. So in order to get around this, we just break these disaccharides back into their constituent monosaccharides, right? Back into their original parts, and then test them that way, okay? A little loophole, but it's cool. So here we have an unknown solution, and I'm going to add my boy Benedict into it, okay? And see if there are any monosaccharides, any reducing sugars inside. I do that, and there's nothing, mate. Absolutely nothing, okay? I'm shitting it. So it's blue. It's remained blue, meaning there's no reducing sugars, there's no monosaccharides, but there still might be disaccharides, okay? They're like these little invisible creatures that we can't see. So in order to do that, we first break them. We add hydrochloric acid into it. We acidify it, and that breaks these glycosidic bonds. Now we heat it close to the boil. Now, if there are any disaccharides in this solution, we've broken them. We've just broken them into their monosaccharides. We've changed them from their disaccharides into their monosaccharides. So now we have two reducing sugars. But there's a problem here. We have a lot of acid, okay? These conditions are very acidic, and that's not good because our boy Benedict, he's not built for that life, okay? He's a bit of a weakling, okay? I can survive acid. You probably could. That's a lie. But this Benedict guy, he can't do it, okay? He doesn't like acid, so it doesn't work in acidic solutions. So in order to remove an acid, think back to your GCSE, in order to get rid of acidic conditions, we need to neutralize it. And the way we neutralize an acid is to add an alkali, a base, right? Something that can give us hydroxide ions, right? So the best thing here we can do is add sodium hydroxide. We add sodium hydroxide, it neutralizes the acid, and it gives us water, right? Hydroxide from the alkali, and it combines with the H plus from the acid. You put that together and it makes water. Now, if you didn't understand a single thing I just spewed out, don't worry too much about that. Just understand that if you break these disaccharides with an acid, we can't just leave it like that because Benedict solution doesn't work. We need to remove the acid. The way we remove it is with an alkali. That's it. So we've done all that, and now we have our monosaccharides. We just test it as normal, okay? We test it, we put Benedict in it, and here's our color changes. So again, blue means nothing. So when we get blue, it means there's no monosaccharides or disaccharides, okay? So pretty much all we just did is we got our sample, we tested it with Benedict solution for any reducing sugars, and nothing came up. So we thought, all right, there might be non-reducing sugars. So these invisible assholes, okay? So in order to test for them, we just break them into their constituent parts, into their monosaccharides, and then test them that way. Boom. That's literally it. And this kind of goes without saying, but there could be mixed results, right? So if you do your first test for reducing sugars, and let's say you get green, right? Meaning there's some. And then you do your second test, right? You break whatever disaccharides there may be in that solution. And then you test it again. and they might be, say, orange. You've just gotten a mixed result, right? You've gotten green in the first one, and then you've gotten orange in the second one, right? So you've got a solution that contains some monosaccharides and some disaccharides, which is a little bit annoying. So overall, monosaccharides, they reduce things, okay? They give us electrons. They're nice people. But disaccharides, they are not very nice people, okay? They don't reduce things. We can't really test for them in A-level biology, okay? So in order to get around that, we just find a little loophole and we break them into their monosaccharides where they can reduce things that way. So just learn that, bro. They like asking you this in the exam. Now, your dumbass probably thinks this video is over, right? But it's not. So just open up your textbook, bro. We've still got a little bit more to go, okay? You need to memorize a few things, okay? And I hate to do this, little bro, but you got to do it, okay? So you need to memorize what monosaccharides combine together to make which disaccharides, okay? Standard simple reaction. So we've got here glucose and glucose, okay? You put these two bad boys together and you create the disaccharide maltose, okay? And of course, this is a condensation reaction, okay? It does release water, but I'm not going to put it in this reaction because I'm too lazy for that. You put these two guys together, they do form water, they do form maltose, which is what our boy looks like here. And you don't need to memorize what maltose looks like, okay? But even if you did, it's pretty easy. You just draw out these structures and you just put them together. You take away your water and that's pretty much it. But I don't think you do. Now here we've got glucose and galactose makes lactose. Now I don't know whatever nerd came up with the name galactose. It sounds like a bloody Marvel villain. And it's quite an easy one to remember because in the name galactose you have lactose, which is what it makes. Easy, bro, let's get it done. Now, the last one here we've got glucose and fructose combined together to make sucrose, okay? Now I have a bit of a silly way of remembering this, okay? But let's say you have a fruit, right? What do you do with a fruit? You suck them. So fructose sounds like fruit, and sucrose sounds like suck. So fructose makes sucrose. Pretty easy, pretty cringe. Remember it whatever way you want to remember it, but, you know, everyone has their own ways. So here I've got an exam question, okay? Now, raffinose is a trisaccharide, okay? This question is for 3 marks. That's pretty much all you need to know. I don't remember which paper I got this from, but have a go at it, and we'll go through this in a second. So let's apply your knowledge here, okay? Monosaccharides, they reduce things. They are reducing sugars. They give things electrons. Disaccharides, they are the opposite. And if that's the case, then surely the same thing would be apparent with trisaccharides, right? And it is. It's the same thing. So raffinose, they don't donate electrons. They are non-reducing sugars. So in order to test for them, we just break them apart. We heat it close to the boil, and then we neutralize all the acid so that our boy Benedict can actually work, because he's a bit of a pussy. So we put Benedict in it afterwards, and it should go orange, green, yellow, or brown, but it won't go blue because the question asks you to prove it. And if it went blue, then nothing's been found. You didn't prove shit. So this is the mark scheme here, and if you got these points, you score all 3 marks. Pretty bloody easy. So that's pretty much the end of the video. Now you can actually close your textbooks. Make sure to subscribe to the video, like all that bollocks, and I'll see you in the next one.